let me just start by number one, we appreciate the opportunity to do this in association with the ART Annual Symposium. We have, I think, a lot of common interest between our two National Resource Centers, and I think that'll come out as we go through this. So we appreciate all of you hanging around at the end of a relatively long meeting. Hopefully this will be a chance for you to interact with us and learn about maybe some of your next steps in your own research program. So let me just start by introducing who we are. So I'm Marcas Bamman from University of Alabama, Birmingham School of Medicine. Rob Model, also from UAB. I'm a professor in cell developmental and integrative biology, which is sort of the regenerative rehab side of my brain and fits in nicely with a lot of what you do. I'm also a professor in medicine and in neurology, which sort of puts me into what we're going to talk about today. Rob?
Yeah, so I'm Rob Model, as Marcus said. I'm a professor in the Department of Physical Therapy, although I'm not a physical therapist by training. And I also serve as the Associate Director of the UAB Lakeshore Research Collaborative, which is a collaboration between UAB and a major clinical setting for the care of individuals with mobility disability.
Yeah, and I should also say that at UAB we have a university-wide interdisciplinary research. I'm gonna feel like I'm doing karaoke up here. So at UAB there is a university-wide interdisciplinary research centers structure. There are 24 university-wide centers. Ours is the UAB Center for Exercise Medicine, which is essentially the home base for this National Resource Center that we're going to talk about. So I direct that center at home, and Rob is one of the associate directors of that center. So when we were asked to do this, we were excited because like the ART AR3T Center, which put on a very, very nice symposium over the last 2.5 days, we're also in this same network of National Resource Centers. And I'll touch on that briefly. Some of you may have visited Carla Perna in the back, who is the administrative leader of this center. We had a booth here with some materials from all of the 6 centers in that network, and I'll touch on that. But the main focus of this is to learn from you sort of where you are in your steps in developing your own research path toward clinical trials. And I'll just start by saying, from the NIH's perspective, all of us, whether you're a student, junior faculty, mid-level, senior faculty, it doesn't matter. All of us who want to do clinical research and do interventional research, in other words, do trials, we all have to step up our game. And when I say that, going forward, the NIH, irrespective of which institute we are all seeking for support, they're looking very carefully at trial design, at reproducibility, at rigor, at resource sharing, at all the things that we all need to do a better job of so that the results of these trials are, number one, more definitive, number two, actually translate to participant and patient care in a way that at this point I think has moved at a pace that NIH has not been particularly happy about. So we're here as a resource center to try to help and guide folks to make that transition to a more definitive clinical trials world. Okay? So what I don't want to do is lecture you for 2 hours. That's not the goal. So the goal is to interact. We have slides to guide the discussion, and I'll show you the outline in a second. But before we even jump into it, just by sort of show of hands, I want to know who we're talking with today. So how many of you are pre-doctoral? How many of you are in a How many of you are in a postdoctoral fellowship? Okay. How many of you are early in your faculty career? Okay. And how many of you are senior faculty? And how many of you are outside of academia? Okay. So you're not sure? You're kind of outside. Got it. All right, so
can I ask one question on top of that? How many of you have actually run a clinical trial? Okay, and it seems like those are the senior people who are raising their hand. So let me just make the comment that when I was at the early stage in my career, I wish there was a center like this in existence, and I wish there were people like Marcas going around, because what we had to do early on is kind of fumble through how to figure out how to design and run a clinical trial, and a workshop like this will really help you accelerate, and utilizing the REACT resource will help you accelerate your understanding of clinical trials. So this is really a great opportunity.
And every time I do this, I learn a lot too, so it's good for everybody. All right, so we are part of a network, as you probably know, some of you know, that the NCMRR, National Center for Medical Rehabilitation Research, with two partnering institutes at NIH, NINDS and NIBIB, supports a network of 6 national resource centers. This symposium was sponsored by one of those 6. We are one of the 6. And then there are 4 others. So there's the Stanford group, which is really a strongly musculoskeletal focused biomechanics group doing a lot of simulation research. Really got some valuable tools and expertise for people who want to model mechanics of a given disease state. The Center for Large Data Research is at UTMB Galveston as its primary home. This is the center to go to for your preliminary data. It's the center to go to access very large EMR data sets and other large data sets, Medicare data sets and so forth, not only for preliminary data, but to actually do epidemiologic research and publish that research. That's a great group. We're the clinical trials group at UAB. NM4R, the National Center for Modulation Research. They're primarily focused on, they're focused on a number of neurologic disorders, but their main wheelhouse is stroke rehabilitation. So if you're interested in really high-tech and sort of state-of-the-art ways of studying individuals with a neurologic disease and intervening on those people, that's a great group. And then, of course, you know what ART is. And then TREAT, if you sort of map yourself as tracking through these resource centers and taking advantage of what they have to offer, TREAT is the final step. You develop a new technology, you now want that technology to get into the marketplace. Most academic institutions, I can't speak for Pittsburgh, I can speak for UAB, don't do this well. It's slow, it's painful, and it's a tough go. But that's why TREAT was created by Rick Greenwald, and they've been around for quite a while now. They're on their 3rd funding cycle. They do a really good job of getting you to understand the steps from, is my idea worth pursuing, market analysis, IP protection, all the way through the chain of events, patenting, and so forth. They do a great job with that. So all those are available to you, and I always like to say, for the skeptics, and all of us who are in academia or in science in general are very skeptical. That's why we do what we do. We ask questions, we want answers. We're always questioning everything, right? So I want you to know that NIH has paid it forward. So every resource that each of these centers has, whether it is consultation, visiting scholarships, pilot funding, on down the line, you're not going to receive a bill. NIH has paid it forward, and we are the conduits of that money. We're the shepherds to try to help you leverage those resources for your research program. All right, this is REACT, and this is sort of what we do. This is the focus of our center. We want to educate people, offer training opportunities for people, so there's a lot of online content. We have a YouTube channel. If you just have a specific topic of interest in your own clinical trials research or in your own development, and you say, I wonder if REACT has a lecture on that or a clinical vignette on that, just go to the website and see. We've got about 60 or so courses or YouTube videos that you can see, as well as a lot of other resources for training and education. We also house a bunch of clinical databases. You might have specific interests. They may not be available in Galveston. We might have them because we also do a number of those sort of housekeeping things. Consultation. So you're an investigator, or you've got a team of investigators, and you're developing a new study design. You're going to write a grant application together, and you get to a point where you're stuck. Maybe it's on page 1. Maybe you need help with the aims. Maybe it's help with the testing of the primary outcome or the recruitment and retention section or whatever it is. We've got people who are really good at this stuff. Gary Cutter leads this effort. He's internationally recognized. He runs coordinating centers for a lot of international and multi-site clinical trials. So he's a biostatistician by training and a clinical trialist. Great resource. Get on the phone with him. Work out your problems. If it requires a visit to us, spending extensive amounts of time with some of the folks, that's available too. Pilot studies. So these centers were not created just as sources of money for everybody to have pilot grants, but we do have that component. And we're about to have another RFA for our pilot program. So we award $40,000 pilots. And we do this every year. And I'll show you a little bit about what we do there. Visiting scientist opportunities, sort of self-explanatory. If you need extended time, maybe you just need to get away. You need to interact with people in developing your grant application or interpreting your data or whatever the case may be, and you can't get away from your phone and your email in your office, and you want to essentially isolate yourself and be very productive and work with other people. You maybe come to UAB for 2-3 days. And then a mobile technology lab, which is really what we're doing under Rob's leadership to try to make sure that when we do community-based and home-based trials, that we do them in a rigorous way where we can collect a lot of biologic, physiologic activity data in ways that, relatively seamless ways. So we're working with a number of companies on technology development, and we're also doing a lot of validation studies of existing technologies for that. And then we have scholar awards. So maybe you don't need a full-blown pilot. Maybe you've got some blood samples in the freezer from a small group of ALS patients, and you've got this question you want to answer, and it would be really nice to get the preliminary data before you submit for the next application deadline. You either don't have the core lab at your institution to do it, you don't have the expertise to do it, or you don't have maybe your core lab doesn't have the time, whatever the case may be. You go to the website, explain what you're trying to do in a paragraph. You might get a scholar award just with an MTA agreement. Send your samples to UAB, we generate the data, send you the data. Okay, so that's also available. All right, let's jump in. So just to give you a flavor, we have 9 current pilots in the first 2 years of our 5-year grant. You can see it's sort of spread all over the country. We have 6 of them outside of UAB. We actually have 3 at UAB, but those UAB investigators are not funded by the NIH grant. They're actually funded by matching money locally. So we let our local faculty compete, but if they get selected, they're on the local matching dollars, not the national money. So we'll do this again. It's already posted on the website if you're interested. Go to that. We're going to fund at least 4 and potentially 5 more pilots this year. All right, so let's jump in. So the intent of this, again, I don't want it to be a lecture, so I want you to ask a lot of questions and interact with Rob and I. So we're going to talk a little bit about design considerations. What are the designs available to you to do a clinical trial? We're not going to get into every single design, and if you're a pragmatic trials person, we're not even going to touch that. We don't have time. So we're really focused more on the RCT model, the randomized controlled trial, and variants of that, not the pragmatic trial today. We can do that separately. We have some experts. Ken Sag is our leader in pragmatic trials design, and Rob actually runs a pragmatic trial now funded by PCORI that we can have a sidebar about that. We're going to talk a little about control groups. What are they? What are they good for? How do you decide what your control group looks like? Stages of development, we'll get into sort of what is a pilot and why do we need a pilot? What's it for? What are our expectations of a pilot? And then we'll get into a couple of really key areas. One I think is really, really important and sort of underappreciated, and that is inter-individual response heterogeneity. What I mean by that is if you're going to design a study of any sort, I don't care whether it's behavioral intervention, device intervention, drug intervention, combination, not everybody's going to respond to that equally. And there's opportunities to do really cool science and advance your, yes? Really? Okay. There's lots of seats up here. Come on over.
He doesn't like talking to the mic, so.
Well, I mean, I'll do this. Okay. But again, don't be shy. And then we'll talk about technology as well. So let's jump in. So design considerations. And again, please stop me, and Rob, you stop me as well. When we are in our center at home, if anyone at any stage comes to us and says, we want to work with you guys to design our next study, this is what goes on the board. Because we want to understand, if Rob and I are asked to, it's sort of a research roundtable format, to work with a group that's interested in epilepsy or MS or Parkinson's or total hip arthroplasty or whatever the case is, spinal cord injury, etc. This goes on the board because this is the crux of the whole thing. Captures everything that can influence your study. It can tell us, we published a version of this in the Mayo Clinic Proceedings 3 years ago. But this has just been modified slightly to sort of add more variables. But we know that there are a number of non-modifiable variables that are going to influence whether or not the primary outcome is achieved and how people differentially will achieve it or not. There's also a number of modifiable factors, and when you design a study, this is what you need to think through. What are all these things all these inputs that can affect my outcome? And then along the way, you've got a number of what I call mediators or modulators. So overall, we want to know what is the phenotype of these people? And when I say phenotype, that's all encompassing. That is how they're being influenced by their own genome. It's also how they're being influenced by their own behaviors. It's also their disease stage. It's everything. It's their phenotype. How many of you, does anybody in here do Parkinson's research? Okay. So if you do Parkinson's research and you have a question you want to answer, can you answer that in an open recruitment of Parkinson's disease, or do you have to dial into a Hawthorne stage? Right? So Parkinson's, if you don't know, is stage 1 through 5. Clinical criteria. It's a progressive neurodegenerative disease. So when we do Parkinson's studies, we're stage 2, 3. Stage 1 is too early, 4 and 5 is too late. So that matters. Sometimes you pick up a paper and they've studied 19 Parkinson's patients and it's wide open. They carry a diagnosis. Well, that introduces so much heterogeneity before they even intervene. And that would be the case for MS or anything else. So that really, really matters. And then, so what's your intervention, right? What are you going to do? What's in this box? And what is in that box is going to be influenced by all of this sort of input, but it's also going to be influenced by what they do outside of your supervision. So this is why, Rob is really focused on how can we translate clinical trials to community and home-based in a much more rigorous way? How can we monitor people and engage people and not just say turn in your questionnaires every 2 weeks, right? Because it matters. What are they doing? And so anyway, this is a nice sort of guide for us. You spend an hour on this and you've written the aims. You've sort of identified your sources, where your challenges are. You've dialed in your population and so forth. So we kind of like to start here. So let's jump into types of design. So the typical design that we are most comfortable with and used to is a standard sort of parallel group design, right? So it's a randomized controlled trial. Enrollment is open to the population of interest. And it might be, Parkinson's 55 to 75, stage 2, 3, Hawthorne. That's the population, but it's open, right? Within that. And randomized to treatment versus control. That's kind of typically how we think of RCT most of the time. But I want to talk about other models. So there's a targeted model, an enrichment design, which might be very useful for certain applications. So this is when I have a biomarker. So we're at the Regenerative Rehab meeting, so biomarkers are kind of a big deal. You're talking a lot about stem cells and molecular pathways and cell signaling processes. Well, maybe there's a biomarker where I'm convinced by preliminary data that one subset of these people, not all I'm going to just stick with the Parkinson's scenario because it's rolling off my tongue. So the the stage 2-3 Parkinson's population, maybe I'm convinced that within that population, I've identified a biomarker, whether it's one of the genetic variants associated with PD progression or whether it's something else, an environmental exposure, and I'm convinced that my intervention is going to be most effective in the target population. May not have much of an effect in the non-target population. This is a design to go for. It's an enrichment design. What are the advantages? It's very efficient. If the differential that you expect between the target group and the non-target group is high, because then you can easily justify a smaller sample size and really have a more definitive study on a small group. It becomes problematic, though, when it really costs a lot of effort and dollars to do the screen to identify whether they're in the target group or not. Okay? Because now you're going to spend a lot of your effort on the front end to bin people target, non-target, biomarker, non-biomarker. Okay? And in order for that to be sort of justified in your mind and in your pocketbook, you better be expecting a large differential between target and non-target. So that's an important consideration. It becomes futile, though, if what you've identified as your biomarker is found in over 50% of the cohort. Now you may as well just recruit the whole cohort. So we're talking about a design that's really focused on that 5%, 10%, 15%, that sweet spot subset. So this is a good design for that purpose. All right, we're going to keep ripping through these, but please ask any questions that you have. So adaptive. This is a popular sort of term, unfortunately misused a lot. We all want to go adaptive design because it sounds kind of cool, sounds kind of sexy. It sounds like we're being efficient with our money, okay? And it's because the trial is incremental. And what people tend to miss sometimes when we talk to folks is that adapted is not the wild, wild west. So adapted doesn't mean, oh, I can just keep changing stuff for 5 years. It's an adaptive design. It's all good. No. The decisions to adapt your trial are all pre-decisions. So it's not along the way, well, recruitment is really going poorly, so we're going to adapt. No. You needed to decide that up front. So access to accumulated data with predefined decisions is the key. So modifying sample size, dropping treatment arms. You know, in rehabilitation research, a lot of times you have a multimodal intervention. So adapted is really good in that sense because over time, maybe you get 20% of your cohort enrolled. That's your pre-decision. And then you determine after 20% enrollment, you've got 3 treatments stacked upon one another, and one of them is not doing much of anything. Okay, you can preset that and drop that. Careful planning is really important for the interim analysis, and this is when you need your version of Gary Cutter. This is where Rob and I would go to Gary Cutter and say, what do we need to do to make sure that we're not taking a peek at data too early, because there are problems with that. So careful decision making about who can see the results and what results they can see to do this interim sort of analysis is really important. And so again, pre-planning is the key. So I'm going to go through some rules. Allocation rules are really important. How do you allocate to the treatments? Sampling rules. Number of subjects you're going to expect in sort of this interim stage 1, stage 2, etc. Stopping rules. Are you using an adaptive design because one of the things you actually want to do is determine whether you're doing harm, right? Is it safe for this to move forward? If you're doing a large-scale effort where you have a data safety monitoring board, they're going to tell you that, right? So they may make you do adaptive even if you don't think you're doing it adaptive. You're not helping enough people, you're reporting too many adverse events, you've got problems. And then decision rules, you have to predefine all your next steps. So this is adaptive in the real world. Unfortunately, we misuse it a lot. And then the last thing I would say is really important. Looking too soon with too small a sample can lead to false positives, major problem, or it will warrant an increase in sample size when it wouldn't otherwise be necessary. So let's say you pre-design your study, you're going to enroll 400 people, and your interim analysis says, you know, we looked at the first 80 people, we probably need to go to 600. Well, maybe if you hadn't looked, when you accumulate that 400, maybe you have what you needed. And so now you've sort of misused this approach. All right, let's talk about this. This is a smart design. Any questions about adaptive?
So, Dr. Bamman, can
Yes,
I
sir.
do what you'd like your audience to do?
Yeah.
So we're thinking about designing a study where we are trying to treat depression in multiple sclerosis. And we're thinking about having people start with either cognitive behavior therapy or physical activity and looking at them halfway through and determining whether or not they respond or whether they don't respond, and then reassigning them to a new condition. Is that considered an adaptive design?
Sort of. It's actually, when you say halfway through, that kind of scares me.
It's a pre-planned switch.
Yeah,
Looking at
but-
after 6 months.
But 50% is a lot. But, you know, essentially what you're asking for is a smart design, which is what we're talking about. Yes?
I guess I have the question of why would you pick an adaptive design? Like, is that because what your interventions are considering are riskier and that's why you're checking in more often?
It's a great question. Not necessarily because they're riskier. It's actually a very practical and smart thing to do, particularly for a multi-site, large-scale effort.
It's more efficient because you can.
It's more efficient to make decisions early. Is what we are doing futile? Do we want to continue to waste the NIH resources for 5 years if after 18 months we essentially determine that this is a futile effort, right? How can we repurpose this, or how can we take a different direction? But again, it's pre-planned. Yes, sir?
Would this also include things like dose escalation studies? Or
Yes, absolutely. So great question. Does this include dose changes or dose escalation? Absolutely. Whether that be drug or exercise or what have you, right? This is an opportunity to take a peek at the data with the right people doing the peaking, and then them telling you as the investigative team whether it warrants you adjusting the dose, as an example.
Marcas?
Yes, sir.
Do people publish native adaptive studies?
Yes. In fact, the question was, do people publish native adaptive designs? And the answer is yes. And you're going to see more and more of it, because what I said at the outset about the new sort of direction of clinical trials at the NIH, I'll say from a prominent NIH program officer, we were told in no uncertain terms, no more small, crappy trials. Okay? Ralph knows who that is. It wasn't Ralph, by the way, although he probably agrees with it. So, yes, you're going to see this a lot, I think. Adaptive.
Well, the reason I ask is you learn as much from, at least I do, from a negative trial as you learn from a positive trial.
Agreed.
How you know what not doesn't work.
Yeah, but what adaptive enables you to do is actually determine that you're undergoing a negative trial without the full 5-year, 100% enrollment, right? It makes everything more efficient. And one thing I would say, I didn't put it on a slide, but if you are doing something where you're going to follow sort of the drug model, phase 1, phase 2, phase 3, phase 4, that is an adaptive design. It just takes 20 years. But really, that's an adaptive design. It's phase 1 safety, right? Phase 2 efficacy, phase 3, you know, getting a little bit more effectiveness and so forth. And so they're doing this along the way iteratively. It's just too slow. So in medical rehabilitation, we have the opportunity within 5 years, in most cases, to do this much more efficiently. All right? Smart design. Let's stick with that. So this is basically a subset of adaptive. And it's kind of going back to the dosing question. This is appropriate for chronic conditions where treatments work but may require variation over time. So in a smart design, as the title reads, sequential, multiple assignment, randomized trial, this is the opportunity to re-randomize people based on how they're responding. You don't want to give up on people, right? We don't want to give up on people. So I blame, every time we do a clinical trial of any size, I know, because I've done the science long enough, I know in the end I'm going to have people that would be considered sort of low responders. And we'll talk about that in a few minutes. But I blame that on me more than them. So what did I do wrong? I didn't give them the right intervention. We're now getting into more precision approaches to this whole thing. Going back to our original diagram, what are all the inputs and mediators that may have resulted in them not doing as well as the person right next to them? So now that makes us as scientists think about how do we optimize the intervention for them, not give up on them. So this is a smart model where you can do that, re-randomize them to an alternative, where you have high heterogeneity. How many of you have ever experienced this in any of your studies? You get to the end, standard deviations are huge, you start looking at the data, and you've got people that range from 0 benefit to 90% improvement. We've all seen it, right? We're not going to give up on the 0 benefit people, are we? I'm not. But we're going to figure out what we do wrong for them. Okay. So you want to focus on the key outcome, the key hypothesis, because if you decide to be smart about it on every potential outcome or every potential question that you could ask, now you're becoming very inefficient because you can't do it. It's impractical. All right. So and in this case, to do a smart, sort of cleanly, these are mostly binary type studies. Either they got the benefit or they didn't. Yeah.
In terms of the re-randomization, how much are you able to look into the way other people have responded to like select? So I mean, you're randomizing them to a new group. You're not selecting them, like which group you want to put them in if they didn't respond.
So
That was clear. I can try to rephrase it.
there's a couple of different ways of doing it. Again, remember this is a subtype of adaptive. So if I'm going to go in with a smart design, it's not something I come up with at year 2.5. We've been dumb for 2.5, now we're going to go smart. It's smart from day one, where I'm already going into it saying, here's my primary intervention, here's my 2 alternatives, and we're going to start with the primary intervention. And when I identify people with a signature of poor responsiveness and a signature that makes me hypothesize intervention B for them or C for them, it's all sort of pre-planned, right? And then you can get away with that, and you'll get data out of more than one treatment that way. All right, but it has to be pretty simple from the standpoint of, you're not looking at gray. Gray becomes a big problem in a smart design, or any adaptive, frankly. You can't be gray. You can't say, well, 20% improvement versus 33% versus no. You've got to have cut points. Did they meet the mark or did they not meet the mark? And that's what makes it binary. Yeah.
Do these types of designs include like tiered treatments, tiered interventions where they like prevent plateau? Because the patient makes a certain landmark, progress the treatment.
Yes. So this is the question, if you didn't hear it, progressive models where you pre-plan progression in a treatment, expecting good response initially, and then they eventually sort of adapt to that and plateau out, and then you're going to change the treatment or step it up or maybe even step it down, right? If it's exercise, sometimes it's actually the opposite, where the benefit comes from these sort of reduced volume and intensity periods, right, and then ramp back up. Yep.
Could you comment on the sample size estimation in these type of designs and feasibility of doing them in special population?
The sample size estimation for this type of design is not going to be all that much different than your standard RCT. Because statistically, we have the tools to study the subsets well and in an unbiased way. You wouldn't be, if you're the PI though, the adaptive decisions and the smart decisions, I'm not going to call you dumb, but I'm pretty dumb. I can come up with a question and a model that I want to pursue, but I don't want to be the one to make the decision about changing something. I want to be involved in the planning of those decisions, but I want a Gary Cutter or somebody who is blinded to everything else to come to me and say, these are the results, change. So from that standpoint, it won't change the size of the study all that much. But if you're in a rare population, as you already know, if you're going to move, so regardless of the model, if you're in a rare population, let's say spinal cord injury, I would say, Ralph, you can either agree or disagree with me, but I would say, and I don't want to scare people, but unless you can really justify a small cohort, the days of a single site N equals 12 SCI study are over. You know, NIH wants to see SCI studies of N equals 250 across 6-8 sites. And that's got to be the new direction. And we all need to start thinking that way.
So generalizability.
Yeah, you want generalizability, not so you want, you know, multiple places around the country. You want to tap into multiple, back to slide one, you know, tap into multiple non-modifiable factors, right? Different people. Yep.
I'd say one of the problems that also occurs a lot is there's, for some of these, maybe not rare conditions, but not necessarily all that common, there are a lot of studies already ongoing in which that limited patient population may already be enrolled in studies. So you have these competing studies for that. And if you're in a single site location, that just really hampers your ability to get that.
That's exactly right. So we have, I can tell you at UAB, our neurology department is one of the highest volume Parkinson's clinics in the world. But that doesn't mean I have direct access to 10,000 PD patients. It means we got 10,000 studies going on, right? So you're exactly right. You got to go multi-site.
So one comment on feasibility of these sorts of designs. The example I gave earlier about switching treatments for depression and MS is a real example. And we were actually engaged in some patient-centered interactions with them, where individuals commented to us, that they were worried about enrolling in a randomized controlled trial because they didn't know if the treatment was going to work for them, and if it didn't work for them, would they be able to switch to another treatment, right? And so in this context, it really turned out to be a very nice design from a feasibility of enrolling subjects into the trial perspective, because, you know, when you take something like cognitive behavior therapy for depression and multiple sclerosis, it works 40% of the time to get that binary outcome. And we know that exercise works about 40% of the time. And so people were really worried about, what if I don't respond? And we said, well, the design we're thinking about using is halfway through, we'll look at whether you're responding and then switch you to treatment if you're not responding, right? And they were very, very excited about that. And I think that's ultimately going to help us do the trial more successfully.
And that mimics clinical practice decision-making.
Absolutely.
Yeah, so that example's great, Rob. I'm glad you clarified, because what you were talking about is halfway through the participant's time in the study, not halfway through the study. So that's a I didn't catch that the first time, but that's actually really important. So if you have a long-term intervention window, you can do exactly that. Make those switches in real time with folks. All right, let's talk about control groups, or sometimes just comparator groups. It's not always practical to have a true control group, right? The reviewer of your application doesn't always appreciate that. So you have to be able to give them, you have to go into this sort of what I call street cred to explain to them why in your study a true control is not in the real world, okay? And that's the case in a lot of our medical rehabilitation world, isn't it? There is no such thing as a true control. You're not going to truly deny somebody of any sort of treatment. So a lot of times what we have to go in with is our experimental treatment versus usual care, right? Now if you go, we don't even need to get into multi-site. Let's just talk about single site. Let's talk about UAB. Let's throw UAB under the bus. If I'm going to do a UAB only study of a given disease and my comparator is usual care, I still have to collect all the data, because from therapist to therapist to therapist to therapist within our own clinic, not our research clinic, but within the medical machine, there's no standard of care, right? So usual care becomes messy, but we all have to accept the reality of that. So Defining the control or comparator is one of the most critical components. This is where you need a really strong PI, team, biostatistician. This is day one. You got the figure up, you're designing the study. After you figure all that out, intervention, population, potential modulators, etc. Before the meeting ends, control group, comparator group, what does it look like? Because until you come up with all that stuff, you can forget about sample size estimation and differential estimation and all that stuff. So you got to do this carefully. So comparator group is not selected randomly.
One quick comment on this is that I think a lot of times people just design a study and say, I'm going to have a control group. And they never really think about what that control group is and what they're trying to control for. And this is, I think if you talk to Ralph Nitkin, he'll tell you a very important factor that they see in grant reviews all the time where people just say, I'm using waitlist control. And they haven't thought about it any further than, well, that's just what I read in a lot of papers and it'll be okay. It really requires a lot of thought and attention to figure out what it is, what you're trying to control for, and then describing it very well in your application, and you will be much more successful over time than just grabbing the first thing that you think will work.
Yeah, so again, great points. This is a careful part of trial design that you have to give the attention that it is due. Otherwise, you will get bitten later. Either from in the real world, you'll get bitten with your data, or in sort of the application, the grant world, you'll get bitten in review. So you need to think carefully about this. So sometimes it's perfectly justifiable to have a comparator group that's not selected randomly and is not receiving the treatment. Okay? Sometimes that's okay, and you can think of examples where it's actually unethical to have a true control. You can't withdraw care from somebody who's progressing along this, progressive neurodegenerative disease like ALS or something, which in some people can move pretty rapidly. You can't say, here's my new and improved treatment, or you have to do nothing. So you have to think about it in that sense. And also sometimes, frankly, it's the key to the comparator is this sort of lack. You've lost the RCT model because you're not randomizing people. But in pilot studies, sometimes that's the way it goes. You just have to have some data that suggests that the direction that you're heading is a good direction. And it's not always possible, feasible, to do a true randomization of 10 people in your pilot. Sometimes you have to, for practical reasons, actually identify people willing to come in and do the treatment versus those not, and you at least have some data. You go in, open book, it wasn't randomized, but these are the results, and we think this is an important difference. So a control group is randomly assigned, as you know. Control group not engaged in the study other than for testing sessions is not optimal, and some people would say is absolutely not acceptable. I think there's a range of interpretations on that. I use the term not optimal because you actually want to capture other things about those people that might influence outcome. Okay? In this case, it doesn't control for possible confounders, behavior, attitudes, perceptions, outcomes. And this is really important. This warning is really important. So participant expectations are known to influence outcome. We've all heard of the placebo effect, right? Which is right there at the top. Outcomes arise from subject beliefs about the treatment rather than the treatment itself. That's why you do the RCT. That's why you do the RCT. That's why when we do double-blind, placebo-controlled drug trials, people have pills that look identical to the treatment pills. We actually have a study like that now, which is a rehabilitation plus and minus drug intervention. So we paid a company to make identical pills. Yes?
I just want to raise an issue that I think particular to rehabilitation, that if you have a study that doesn't have such a sort of null control as one of the control groups, then you run the risk of really testing two interventions that are both effective, finding their difference, and then missing out on actual estimation of what the effect is of this type of intervention.
You're exactly right. And I will, depending on the scenario, I will argue in favor of as much of a true control as we can get. And there are cases where I would argue against it. It really depends on the given test, the given study design, the given population. But you're right. I can tell you, in what I have fallen back on may not be the best answer for every scenario, but I continue to fall back on usual care. Because if I was to make that group more strictly controlled, just as you just said very nicely, you're introducing another intervention. So you do have to sort of walk a line. You don't want to necessarily be criticized for testing 2 interventions. But I think the point of this is you can't just say, oh, thank you very much, you're randomized to control, I'll see you in 6 months. What this is really speaking to is some level of engagement with those folks. Did you have a question?
As much a comment
Yeah.
as a question. I think that the difficult thing for all of us, and we all know this, is from the moment you conceive of a clinical trial and
Yeah.
know your preclinical data, whatever, to the moment you start your clinical trial, it could be 5, 6 years. You got to get the clinical trial funded, and then by the FDA and the IRB and HARPA, blah 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 blah. We just spent 4 years with this. But one thing that's really disrupted the results of a couple of very expensive trials in the last year or 2, was that, and again, it points to the absolute importance of what you're saying here with control or comparative groups, is in that time period, from the time you put your trial together to when you started, it could be 4 or 5 years, the observational control treatments or whatever they do, they can have migrated and improved, and so that you need to take, before you start, somehow take a fresh look at that observational control.
Yes.
Because if you don't, and it has migrated, something is improved, all your power, you're gonna have, if you don't readjust your power for that, it's a great point. A null result in 5 more years and $20 million.
So I think it's a really good point. I want to just reiterate it because I don't know if you all heard it, but because it takes a while to get something started, a lot of times we're studying old ideas by the time we do them. The point is, rehabilitation is going to continue to evolve. And so what you thought was usual care when you wrote the application may not be usual care anymore when you get it funded. So you need to be current. You need to be ready to make adjustments to your design based upon the current version of that control.
And I'll just say one thing as a fun illustration, is that a study just got released today, it's in the papers, in cardiology, showing stenting or not.
Yep.
Not stenting may not be, you guys rock the whole foundations in cardiology.
Yes.
And it's because they did a true control.
Well, yeah, absolutely. Well, about 5 years ago, it was demonstrated very definitively that exercise outperforms stents anyway. But they didn't listen. Maybe this new one they'll listen. All right, excuse me. 2 more important warnings. The Hawthorne effects. Subject alters their behavior as a consequence of being observed. How many of you have ever tried to do dietary intake monitoring? Every 3 weeks we're going to have you do this 4-day diet record. Put it on the fridge, write everything down that you eat, take pictures of it, etc. How many of you get back broccoli, grouper baked, right? A couple of apple slices. So people, particularly people who are enrolled in a trial, they now see this, this intimidating Dr. Model and his team as the authorities, and they want to please them. Not everybody does, but some do. And so that's a really important thing. You got to understand that this does happen. The Pygmalion effect, subjects perform at the level they believe others expect of them. This is a big deal, particularly in rehab. Exercise, right? So we deal with this all the time. Levels of free living activity versus supervised intervention. You have to know what your usual care or your control group is doing in the free living environment, because they know they just got booted. You know, they wanted the intervention, they didn't get it, and well, Dr. Model's team is studying this intervention because I have MS and they must expect that this intervention is going to work, so screw them. I'll fill out the questionnaires for them every 3 weeks, but I'm going to do this thing. You know, so you got to be careful about that and try to figure out, and that's where biosensing comes in and really kind of understanding what these people do. All right, continue on the control group for a minute. Study outcomes are most robust when the control group is actively engaged. We talked about that. That's actually important. And this really has to do with, you know, walking a line between intervening on these folks and making sure they're engaged. So there are ways of doing this. The active ingredient, you know, in medical rehabilitation has become sort of a buzz term because there are so many multimodal interventions. You want to get to the end and say, what was the one that did the work? You know, produced the outcome, and that would be called the active ingredient. So if you can basically give a control, if you're confident my active ingredient is X, so my usual care group is going to be, you know, exposed to almost everything but X, then I'm really testing X. But if I think my active ingredient is X, and the usual care group is going to be restricted from A, B, C, D, E, F, G, H, all the way to X, now what are you really showing? All right, when the active ingredient is well isolated, there still may be factors that confound. So was the dose of the active ingredient sufficient? That's a problem. That's where your pilot study comes from. Okay? Did the subjects attend? Adherence is a huge deal. It's not quite as big a deal in drug trials, although you have to still do careful monitoring. It's not sufficient just to have, say, bring me the empty bottle, right? We actually I have a co-investigator at another institution I'm working with now who developed a very nice little app, IRB approved, little videos of pill consumption at home. That's pretty extreme, but you know, you can go down that type of road now. But when it's a behavioral treatment, I'll stick with exercise because that's what we love to do. Your adherence rates really matter, right? You've designed this intervention. You said, man, 4 days a week of this intensity, et cetera, this is what I know is going to work the best. What are you willing to accept? A 2.5 days a week average? So adherence, or you know, when you get to your responder, non-responder analysis, oh, it turns out they're actually not different phenotypically. It's just that this group came two days a week and this group was compliant, right? So you got to be careful about that. Were subjects immersed and engaged? Did they develop skills over time? Did they use or practice the new skill outside of the training sessions? And that sort of all speaks to monitoring these folks. All right, alternatives to the sort of the classic. Yes.
So let's take a second and think about this from the more junior folks in the group, not those who've been involved in randomized controlled trials. How are you going to design your control group? What are you going to do if you have a rehabilitation intervention? How are you going to select your control condition? This is the audience participation section of this. Yes, ma'am.
So you have to look at what you already have. So you have to look at the population you are already considering as an intervention group. So say it's all of your inpatients or whatever. And if you're looking at specific diagnoses, well, more generally, like you can say, okay, everyone who's coming in who had a spine surgery. So you could look at that as being, this is the pool of patients that I want to target, and then narrow it down to who do I want to expose to the intervention versus who would be a good control person. Either they're ineligible for some other reason, but they would be exposed to the same therapies, the same unit.
Okay. But the key is going back to what he said, is defining what the usual care is. If they're receiving usual care as your control group, if you're using multi-centers, getting a therapist that will be participating, having them all on the same page of what is your usual care. How are you going to treat these patients? Maybe even having a little bit of an established protocol that they maintain some feedback consistency.
There's one, I'm not pushing back because I totally agree. I think that's how it should be, philosophically. But for one of our ongoing trials, which is a post-surgery rehabilitation trial, we were forced to accept the variability of usual care. Because if I tried to create a CRF for usual care and standardize it, I've now got a second intervention. I would love for that to be standardized, but we just, you know, you got to be, it's just a fine line. Yes?
Is that like unclear control group care that's going on? Is there any sort of information that you can get from it to, I don't know, be aware of what's going on, or is it kind of just?
Yeah, I mean, we do monitoring, okay? So in-home logs that they complete for us, logs that they complete when they go to their physical therapy clinic. We know, at least to the best of our ability, without monitoring them electronically 24/7. See you, Ralph. Okay, thank you. Talk to you soon. We do our best to know what they're doing, and we accept the variability inherent in that, and it's really unfortunate that it is so variable. You would think by now the therapy community would have what they considered ideal therapy post-total hip arthroplasty. No. Yes?
I think things like that have been done for like fall prevention. Probably one of my papers that I appreciate was looking at plyometrics or jumping training for older adults. And so they made a group balance class, which look at like this is sort of like well-established Otago program, or has all the key ingredients that clinicians would agree would be a good balance exercise program. And then with the comparison group, they just took 10 minutes out of the more active phase of it and put free self-paced jumping. I mean, just like, we're not talking like box jumps, but just like little jumping jacks or things, just where they were coming off the ground, and they compared those two groups. So it was like, that was the secret sauce, or that one piece that I thought was a really sort of reasonable comparison.
And it's really a nice example because you've done a number of things in that. You said it's group exercise. Yeah. And so anytime you do group exercise, what do you have going on besides exercise? Social interaction, right? And so in that context, you've now controlled for social interaction. What's another thing that's going on in supervision? You're having attention paid to a research participant. Does having attention to you change your behavior? Absolutely. So now you've controlled for social interaction and the attention paid by a supervised individual. What does this now buy you? It gets you closer to being able to determine whether or not your intervention was actually the thing that changed the outcome of interest, instead of being, subjected to alternative interpretations, social interaction, attention being your agents of change. And so that's a perfect example. And that's what I just want to go all the way back to something that Dr. Bamman said earlier. At REACT, we're here to consult with you. We're here to actually have a dialogue and a discussion to pay it forward to help you figure out what is the best control condition moving forward. So please do contact us, do get in touch with us so that we can help you walk through that decision.
Yeah, for sure. All right, I want to continue to interact and make sure we answer all the questions. I do want to try to get to a couple other topics, though, so we don't run out of time. But please keep stopping us. This has been great. So let's talk about alternatives to sort of the classic randomized control. Rob already mentioned this earlier. Delayed intervention. How many of you have ever done a delayed start? This is a great model. In some cases, this is a great model. So now, when you do your randomization and the person, it's revealed to the participant that they got the control group, it's not the end of the world. You're going to get the intervention in 16 weeks. I'm sorry, but please be patient with us. This is a great way to get to
one of the problems we've had with the delayed intervention are drop-ins. So people dropping into an exercise trial. They know that it's not exercise, and they all of a sudden join in another exercise group, right?
Yeah.
While they're in our
yeah, they don't want to wait the 16 weeks. Yeah, they look. Well, yeah, we've dealt with it too, and that goes. A lot of these things we're talking about lead us right into the remote monitoring of people, which is, we're not there yet. It can be sort of pretty, kind of invasive, but if you've got somebody enrolled in your study, we want to, and we're working with companies to try to do this, we want real ways, real practical ways of just sort of tracking what they're up to and communicating with them too, not just secretively tracking what they're up to. All right, crossover design. Great design in some cases. Anybody ever done one of these? So now you're either testing, you've got treatment and you've got control, or you've got treatment 1, treatment A, treatment B, and the same people do both. The order in which they do them is the opposite. So they're randomized to which one comes first. That's a great model sometimes. When would that, well, let me stop. When would that be a bad model? Can you think of an example where a crossover would be a bad thing?
Read some studies where you don't have enough washout time.
Exactly. It's the washout. So let's say it's an exercise treatment. I'm stuck on my favorite thing. There are physiologic adaptations all on different time courses of detraining, right? You're interested in the neurocog. You're doing fMRI, you're doing hippocampal volume, you're doing functional connectivity, whatever you're doing. Has anybody really roadmapped the time course to return back to baseline? Cardiorespiratory fitness. Yeah, we kind of understand when mitochondria start to degrade in muscle. We really don't have any concept of the reversal of training effects in the heart and lung. Okay, you get where I'm going. Crossover becomes dangerous when you don't know the washout period. Okay? If it's drug, chemically you can measure the washout because you know the half-life of the drug, you can assess the metabolites in the blood, etc. But you still don't know whether you washed out the effects of the drug. You've washed out the drug. So it can be dangerous, but it is a really cool model if you can demonstrate that you understand your washout. Then there's a run-in or sort of a wash-in design. Everybody participates in an initial period where they're in the placebo. This is actually a really effective model. It can be efficient. So now we've got everybody enrolled, and we're going to do repeat measures for 6 weeks. And over those 6 weeks, everybody is considered a placebo participant. And in doing that, we're going to get some good data, stability of our outcomes, and so forth. And you'll have essentially your data for comparison. Useful for assessing stability. I just said that. All right. And you may also have trends in change. How many of you do strength testing for anything? Okay, you teach somebody how to give you maximal effort on a strength test. First day, are they giving you maximal effort? They don't even have a clue how to synchronize motor unit activation. If they've never done maximal effort in their lives, you expect a strength test to be meaningful? No. Multiple exposures before you actually know their strength voluntarily. You can E-stim them, right? Which we do. But E-stim in a relaxed muscle is not very comfortable. E-stim in a voluntarily activated muscle, you don't even know it's happening. So you can always do that and look for force deficit, but first day of exposure, no way. It's a pointless measure. And when you see in the literature that somebody's intervention improves strength 270%, that means their baseline data suck. So that's just an example. If we were doing repeated exposure, that's on video, isn't it?
Now we're getting technical.
But it's really important because measures like that have a learning component or an adaptation component will change over the course of that wash-in, and you need to understand that. N of 1 randomized design. This is kind of interesting. Gary Cutter loves these. I'll talk a little bit about these. This is an interesting approach. It doesn't mean you got a 5-year, $5 million grant, you're going to enroll one person. What it essentially means is everybody's their own control. So in an N of 1 design, repeat observation across time in a subject, okay, with intervention effects being reversible upon withdrawal of the intervention. So this is only useful when you know the washout and when you know that your treatment is essentially an on or off model. And if you do know it's an on or off model, you can collect serial data on and serial data off, and that's an N of 1 design, okay? And it's really robust if you understand the condition is stable and the intervention is rapid. Rapid up, rapid down. If there's residuals, first of all, if it's a dynamic condition, so you can't do this in an acute TBI, okay? But you might be able to do this in 25 years post-SCI. You're not going to be able to do it in the first 6 months post-SCI, but 20 years out, yeah, okay. As an example, potentially. And okay, so a specific subtype of an N of 1 design is called a multiple baseline design. Now you got intervention effects are not reversible, or in situations where the intervention should not be withdrawn. And this would be a special case where you've got something that's really helping people and it's actually not ethical to take it away from them, as an example. Or you have an intervention that actually causes some, I hate to use the word permanent, but a semi-permanent change, and you can't go backwards, then this is a model that can be done. Major distinction of these from traditional, all subjects eventually receive the treatment. So there is a lot of merit in that. You're not, that participant is not doomed. Some people will be perceiving this as doom. Man, come on. I got stage 3 Parkinson's and you're telling me that I got to do nothing basically, or do what I'm doing now. And I was hoping for this really cool study. Yeah.
For the first sub-bullet for the delayed
Yeah.
intervention, do you give the delayed period the same amount of time as the intervention period so that you have like a middle time point, I guess?
Ideally, yeah, but it's not always practical either. So I've seen it where it's truncated. Really what you're looking for is stability of measures, but ideally if you can make it symmetrical, 12 weeks, or whatever, it's perfect. But let's say that your intervention to affect change you think is 18 months. Then a delayed start of 18 months is not realistic. So you kind of just have to see where you are, right? All right, stages of development. Now this is where the rubber sort of hits the road. Let's talk pilots. Rob and I talk a lot about pilots and help people with pilots, think about our own pilots. Everybody's doing pilots. Doesn't matter where you are in your career, everybody's doing pilots. So how much data are enough in a pilot? That's the big question. We have a pilot grants program. We just advertised it to you. I hope many of you seek it out. And what I will tell you right now, if you get on our informational call, you're going to hear this loud and clear, so I'll just tell it to you now. Don't design a pilot to answer your question. There's no need for your RCT now. So that's not what the pilot is for, and a lot of people fall into the trap. 25-year, not pointing at Rob, but just using it, somebody who's been in the game 20 years, 20 plus years, right? And somebody who's fresh out of school can both fall into the same trap. All the reviewers, all that panel of people, they got to see a power calculation, blah, blah. If you're showing them you could have done it in 12 people, why are you telling them you got to have 75 people, right? So, main goal, to assess feasibility and avoid potentially disastrous consequences of going into the 5-year effort blindly. You could have potentially drowned the whole research effort, and you could have essentially just avoided the whole thing with a simple feasibility pilot. We've done this. We're doing it now. We're actually doing a pilot in head and neck cancer rehabilitation, concurrent with chemoradiation. And I'm learning, I'm sort of on the edge, is this going to be feasible or not? The intervention works great, but feasibility goes beyond whether the intervention's any good. It's also, can you get these people to show up? Can you get them to enroll? They're thinking about life and death. Are they really going to care about this study, right? Feasibility becomes a really big deal, and that's why we do these pilots. All right, never test the depth of a river with both feet. That's all you need to know about a pilot.
So I think one other thing just to bring up that people don't really think about when they think about pilot is feasibility data relative to how much time is it going to actually take you to deliver the intervention. How many resources do you need to deliver the intervention? How much does it cost to deliver the intervention? All of these things are also important. How much effort do you have to put into recruiting people for your intervention? This is also part of the pilot study framework, because these data are just as important when you go write a large application to justify the amount of money you're going to put on recruitment, the number of personnel you're going to put on a grant relative to get the work done, the cost of the equipment that you need or the facilities that you need access to. So when we talk about pilot, we don't want to just talk about, we want data to show that maybe our intervention does something. There's some signal there that's greater than noise. You also want to think about all the pragmatics that go into doing the research itself and gaining those experiences, so you can very eloquently and from an informed manner write about that in your larger trial. And I think it's something that people forget about. And in fact, there's an entire journal nowadays called Pilot and Feasibility Studies. So not just does it work, but everything that goes into making it work.
And now you can publish it.
And now you can publish it. But there are really good guidelines out there on how to do more than just collect your outcome data, but also your feasibility data.
Yeah. So other goals and benefits, Rob just hit on a lot of these. You want to have some hint that you're on the right path, but that doesn't mean the full-blown sample size estimation and the demonstration that you've already proven that this works. You're going to have some hint. You want something that can allow you to estimate a sample size, but you don't want to say, you know, I've already got p equals 0.02, right? Why do it? To test safety, that's a big one. You want to enroll enough people, not a ton of people, but you want to know you're trying some newfangled device. You know, is this okay? Are people getting hurt? Are people getting, you know, aches and pains or whatever the case may be? So that's an important one. Feasibility, as Rob just said, you can do what you say you can do. When you write a grant application, that is got to come through loud and clear. The aims are number one. Is it innovative? And the next thing, can they do it? Is it important for the field? So is it significant? Is it innovative? Is it going to advance the field? And then, is it feasible? Can they do it? Is this the right group to do it? Sample size estimates, I just said, that's obviously one of the reasons we do them. And that's the reason most people think we do them. And it's a big part of it. But you also, as Rob just said, test and tweak recruitment and retention strategies. Really, really important. Sometimes we go into these things pie in the sky. We're going to enroll 100 people. The clinic says they got 5,000, you know, patients per year. No problem. I can enroll 5% or what, you know, right? Easy. Well, maybe not. Maybe you spend 6 months trying and you're getting, you know, 1 out of 50. Oops. Something you need to know up front. All right, let's sort of jump away from clinical trials design. A couple more topics to introduce you to. I want to go kind of quick and again ask as many questions as you have. So this has been a little bit of a thread today, this concept that not everybody responds the same. I call it inter-individual response heterogeneity. So why do we care about it? Well, it matters because it influences your sample size estimation, it influences the power of your study, it influences who you're going to recruit. So, what's the value? And we're going to talk about that. What are the sources, strategies to actually leverage it, okay? And then we'll talk a little bit about kind of the future of it. So let's just jump in. Example from us. You want to improve cardiorespiratory fitness in a group of people, VO2 max in a group of people. You're going to say, oh, we got a 15% improvement in our study. Those are the modest responders. Awesome. But you got 34% of these people with nothing, and you got this really interesting group that all improved over 20%, which we would call high responders. So wouldn't it be cool to understand what makes them tick and what makes them tick? As opposed to just, where's the mean? Now we're trying to gain muscle or restore lost muscle in aging people who have suffered a lot of atrophy. So a high intensity resistance training, same thing. Same intervention, everybody's stuck to it. We don't have adherence differences, but we get these people who spend all this time and effort, don't get much out of it at the fiber level. These are muscle biopsies taking and looking at muscle fiber size. But what do? We report in the literature, well, they gained 35%, pretty awesome. But we ignore these people, and now we've got, wow, look at these really interesting people. Huge gains in muscle fiber size. They all started the same. I want to know what underpins that. What are the sort of molecular underpinnings of that, the behavioral underpinnings of that? It's really kind of a direction that we don't think about in clinical trials. And I wanted to bring this to you all because particularly the meeting that we're all attending here, Regenerative Rehabilitation, where we've got a lot of sort of molecular minds walking around and thinking about things, this is what you can do to add value to any trial. So even if you're not the PI, you identify a study on campus that is recruiting and intervening on people in a rehabilitation setting, and you know, because I'm telling you this, and everybody, people who've done these know it, you're going to have response heterogeneity. That investigative team may not be appreciated or think about it. You can add value to that study tomorrow by saying, I want to study their heterogeneous responses and dig in. Can you give me some blood? Right? Can you give me their behavioral data? All of a sudden, you've got a really, really nice ancillary. So what's the value and what can we learn? Studies focus on the mean. As I already indicated, variance is overlooked. I think it facilitates precision approaches because we dial into what makes people different. So I think it's really, really an important approach. I mentioned this earlier. We don't want to give up on the low responders. We want to change our approach to those people. So again, back to our diagram, I put it up as the study design diagram. It's also defining all those sources of heterogeneity, which becomes really useful for you to think through it. Where am I going? What am I going to study? What do I have available to me to study? As I talk about this at a lot of these types of meetings, sometimes you get the critic who says, well, the NIH isn't going to just willy-nilly give me extra money to collect blood that I have no aim for. Well, actually, yes, they will. Because they know they don't want to repeat this 5-year trial just to get the blood. There are creative ways for you to add that on, for sure. All right, so this is specific to rehab. So this would be any treatment, all the different sources, at least that we've come up with for response heterogeneity. And now I'm talking rehab-specific types of things. Specifics of the condition. For a traumatic injury, site of injury really matters, right? It's not enough to say I'm enrolling TBI. What parts of the brain are affected? You can look in the literature and see papers that show major differences in outcome. And when they dig in, they are, oh, wow, these are all prefrontal cortex. Oh, these guys are, somewhere else. So you gotta know where that is. Duration of time. I'm not going to show a bunch of papers today because I knew we wouldn't have time, but I'll just tell you, duration of time since injury is a huge one. How many of you have ever read a paper in something like spinal cord injury where enrollment, when you dig into it, you see that it's, 6 months to 25 years post-SCI? Are they the same? Probably not. So that matters as well, and that's a really important one. In fact, there's some really nice work out of the Cleveland Clinic in stroke that shows the plasticity of this rehab intervention that they have. It matters. It's all about the first 6 months. So those people that they enrolled that are 3 months post-stroke or 3 years post-stroke got nothing out of it. People that they enrolled within the first 6 months, big improvements. So that matters. Chronic disease, stage of disease, we talked about this as Parkinson's as an example, duration, etc. Really important to consider. You got to dial in on your group. Or if you want to go big and you want to do something that's much more of a sort of pragmatic effort, large scale, all comers with MS at all stages, fine. just recognize that you will now have a big data set to dig in to things like this, duration of disease and so forth. Post-surgical rehab. Any of you do post-surgical rehab studies? So we have a couple of efforts. One, I already mentioned hip and knee arthroplasty. The other is liver transplant. So liver transplant, as an example, the approach to the surgery really matters. Different surgeons do things different and affect different structures different. In total hip, some surgeons take an anterior approach and some take a posterior approach. So all the surrounding structures, ligaments, tendons, muscles that are affected by the surgery are entirely different. So you're now going to intervene on those people, and you got to know what you got. All right, so we've done some things using what's called k-means clustering, which is what I showed you on those graphs, which enables you to bin these people and understand. Yes?
Probably just
Yeah.
so the message there is, so can you admit like someone who's had the surgery and then go back and factor out those variables? Or should you design a study that eliminates all of that very specific splice because the recruitment issue?
Yeah. I think it's a judgment call. How generalizable do you want it to be? How feasible is it? In the case of a surgery population, I would say probably in most scenarios, you're probably going to want to take the variability on the surgery side and just covary for it in your statistical modeling to understand whether it did have an effect. And maybe if you are doing a pre-planned adaptive, right, and you say, when I accumulate 20%, I'm going to compare anterior versus posterior approach patients, and then your biostatistician is going to tell you if there's a big difference, time to pick what you're going to do going forward. You know what I mean? All right, so omics, we do a lot of molecular mapping. I'm sure some of you do that too, to try to understand what makes people different. Targeted studies, we also do that a lot. We do stem cell biology, which I know is a lot of this meeting, which you can do from human samples. We do it from human muscle. And then behavioral environmental phenotyping, which is really something that Rob does a lot of in multiple sclerosis. That may be just as important as a source of response heterogeneity as a molecular signal that I would be interested in. So all of those different things can be done. Why think about it up front? It has a huge impact on your design. When you talk about effect size, part of what's in the equation is the variance, right? So it's built in, but not, you want to generate an effect size and you say, well, if I get to a 0.5 or whatever the case may be, that's what I want. But part of that determinant is the variability between people. And so you need to know this up front, and you need to leverage it actually to your advantage, I think. Because now you can go in and say, this is what I expect the mean to be, but if I really want to understand the low and high responder groups, I want to recruit this many people. Sometimes that can lead to a lot of really interesting science. So maximizing yield is really important, and I think that message is clear from the NIH and other agencies. We all have to figure out how to maximize our yield when we do these studies. You can't just, spend multiple millions of dollars to say that exercise makes you feel better, right? In a group of people, you got to have more science than that. All right, so evolving technologies, lots of things that we can do with samples and analyzing people. We're in a really good position scientifically now with where the field is in terms of our available tools. A lot of these profiling approaches are getting cheaper and cheaper. We're doing, whole genome sequencing and RNA profiling for very little now in cost relative to what it was 3, 4, 5 years ago. So you can get away with a lot. And then I would say the last thing is this enables all of us to share resources better. And that's a big push for NIH. It's not yet a scorable criterion on a grant application, but it might be soon. I don't know. They are so hot on this that it wouldn't surprise me if it became part of the scoring eventually. Right now we have to write the section, but it's not scored. But if we're all doing this and being sort of forward in our thinking and thinking ahead to what maybe others could benefit from with our study, and we have valuable resources to share, this will be really important. Yes?
You're making me think of resources as well, and I wanted to hear your thoughts on national databases and sort of patient registries.
Yeah.
AAPM&R is trying to pair up with one of the neurosurgery registries and create a back pain patient national registry. And of course, with the huge database of patients from multiple centers, that's exactly kind of what the NIH is looking for. So, are they allocating resources to support that effort, or are they already sort of plugged into national organizations like that?
So there are, each NIH institute kind of has their approach to this. So NCMRR, which is where, a lot of us get our funding, at least some of our funding, they have what's called DASH, which is a data repository. NIH also has biobanking relationships with different groups to do long-term storage of samples that you can access. There's a group in Vermont, University of Vermont, that's essentially a freezer farm for a lot of NIH studies. They have like 200 -80s and really nice data repository, biorepository. So there are ways of tapping in. We in our own, we have a network of 73 sites we call National Exercise Clinical Trials Network. We have a database that tells us who has what that they're willing to share. And so people are thinking about this. It's really important, yeah.
So Marcus, would our partner CLPR?
Now they're data only.
They're on.
So they're a great source of data, but they're not a biospecimen resource. There are others that do that. And we do that too. So even like for REACT, if you had a question that needed to be asked on, serum or muscle from people of certain ages or with a certain disease, we may have it, we may not, but you can always ask. We have a big repository. Leveraging technology. Rob's going to lead this part. We've got another half hour, but keep the questions coming. Okay? Yep.
So, am I on? How about now? All right.
Here, just use this one.
No, it's off. There, I'm on now. Now I'm on. Good. All right. I just want to take a moment to further Marcus's argument for response heterogeneity. We actually had a funded trial where we were looking at the effects of exercise on ambulatory outcomes in people with multiple sclerosis. And we got done with the trial, and we did our analysis of our data, and we found nice mean effects. And so we sent it to a journal. Maybe Marcus was the reviewer, maybe not. And we received feedback. We'd like you to plot all the individual variability, or the individual. We'd like you to plot. I'm going to take Marcus's microphone. We'd like you to plot all of the individual data points per condition. And when we did that, we found something quite profound. Within each group, we had responders and non-responders. And it was really nice because the non-responders, we could actually very clearly classify who they were. They were a specific level of disease status in MS. What's the take-home message? We wrote an entirely new grant just on those individuals, and it was funded, and we had to figure out a whole new program for them, but we figured out a program that helped them work. So to get better in their ambulation. So the point is that this can be really interesting to look at your data. It might lead to new opportunities, to argue for new funding, and development of entirely new programs that you'd never thought about.
That's an enrichment design. You backed your way into it using heterogeneity.
Absolutely. So I'm going to talk about technology. And when I look around the room, I see lots of phones out. Who has their Fitbit on today? Anybody with a Fitbit on? They're falling by the wayside as we speak, I guess. But nowadays, everybody is using some sort of technological device. Your phone can capture lots of data. Carla has an iPhone on that can capture lots of data. And you know, it's so pervasive that it's going to make its way into clinical trials, and we have to be okay with it. And so I come at this from the perspective that the majority of the research that we do is done in a clinical research facility where we have individuals come to our environment. Maybe it's the University Center for Exercise Medicine at UAB. They come in and they have access to very specific exercise equipment. They have access to an individual who's going to very specifically prescribe their exercise regimen. They have access to someone who's going to sit there and monitor what they're doing and going to encourage them and supervise them to do this. And it sounds like a very rigorous research designed to show that exercise has effects. The problem is that those designs are hard to scale and to reach large numbers of individuals. They're also very, very expensive to conduct in a research facility. And so what we've gotten interested in is transitioning from center-based supervised trials into other environments. And if we look at this picture right here, this illustrates that we can very easily do exercise trials in someone's home. We have a picture of a gentleman running on a treadmill. There are other pieces of equipment as well. These pieces of equipment can be seamlessly integrated to a centralized server to capture the data from an individual's exercise session. This individual has a television right there. We could be delivering coaching sessions to help supervise the exercise right in this environment through this really nice television if we turned off what appears to be a Cleveland Browns football game. We can do lots of different things, and this is probably going to help us scale our interventions, reach more individuals, and ultimately have greater impact. So we want to overcome some difficulties with supervised settings. We also want to think about rural locations. So we're at UAB, and UAB is in Alabama, and Alabama has a huge rural community. These are individuals who might not have financial means, or they might not have means to transportation to come into our environment. They live in a rural setting. We can't get them to do our supervised exercise program, so we have to bring it to them so that we can reach that segment of the community. We also have situations with rare conditions. So we talked about issues where we have a very small group of individuals who we might be able to enroll in a trial. Well, if we can deliver this outside of a supervised setting, we might be able to get more of those individuals to engage in our research study. Yes, sir?
Yeah, I just wanted to bring a point that although the circumstances that you identified with rural settings are really true for urban settings too. There are people in certain conditions that will not be able to get to certain areas. I mean, I live in New York, and I can't get people to go to Manhattan. From Brooklyn to Manhattan. I really can't. You know?
It's a great point, and I used to be at the University of Illinois at Urbana-Champaign in the middle of corn and soybean fields. And so we tried to do our research at the University of Illinois in Chicago, thinking, Chicago, great access, right? We had more problems getting people to show up to the research setting in Chicago than we did in Urbana-Champaign. It's a great point. So just briefly, when we're thinking about this, there are two terms just very quickly to review. Telemedicine, which is medical information for the purposes of providing medical care. And we're talking about technology for the remote exchange of this information. I'm going to focus a lot on telehealth, telerehabilitation, which is the use of technology for remote monitoring, exchange, delivery of interventions for optimizing health and recovery. And much like Dr. Bamman, I'm going to talk about exercise because that's what I know the most about. But anytime we want to interrupt and think about a different behavior to apply this to, I'm happy to do that. So what do we need? Well, step one, we need monitoring devices. We need some sort of tool to be able to monitor what someone is doing outside of our setting. And this is a paper that we wrote on wearable biosensors to monitor disability and multiple sclerosis. But you can instrument people and capture a lot of information. And so depending on where you place the sensor, we might be able to get information on activities such as the steps they take during walking or engaging in jogging or running behavior. We might be able to look at falls and near falls and things along those lines with these sorts of sensors. We might be able to look at tremor and how our interventions can affect that with sensors. We can look at sleep patterns. So we can look at awake hours versus sleep hours and kind of describe that based on the quality of sleep as well as just the total volume of sleep. We might be able to look at other vital signs such as heart rate, temperature, and oxygenation. But one of the critical things that becomes apparent when we think about technology is how well our technology works. There's no reason to apply technology to measure something if it doesn't actually do a good job measuring that behavior or that variable. And so what this has done is it's created kind of the look at monitoring systems. That is recognizing that no single monitor is going to be perfectly effective for making an inference about some entity. And so this is a recent paper in Nature Reviews Drug Discovery where they're talking about using a variety of different sensor data, such as motion detectors, location tracking, load cells, and bed sensors, utilizing those in a way that they can make inference about assessments of different things such as gait velocity, location estimation, sleep, and turn that into an inference. And so one of the things that's going to happen in the very near future is figuring out how to put multiple sensors on an individual at the same time so we get a better understanding of what's happening within that individual. So let's think about accuracy and precision. So when we talk about accuracy, we're talking about, do you get a number that represents the number that you expected, right? So if we expected you to walk 500 steps, does our motion sensor actually indicate whether or not you walked 500 steps? And so we can look at this on a group level and we get a group mean that tells us, indeed, our sensor captured 500 steps. But the other thing you have to look at is the precision. That is, sometimes the mean can be deceiving. You can have a device that misses 20-30% of steps in one person and overestimates 20 to 30% in another individual, and when you look at the mean response, it is no difference. So you also have to look at the precision. And these are data that we published on the accuracy and precisions of smartphone applications and commercially available motion sensors in multiple sclerosis. And what we did in this study is we just had individuals walk on a treadmill for a set number of steps. In this case, it was 500 steps. And we instrumented the heck out of them. We put devices on their hips that were very inexpensive, such as the DigiWalker spring-loaded pedometer, through different motion sensors, as well as looking at smartphones. And what you see here is this is the mean number of steps we were trying to detect, 500, and these blue dots in there would be the mean number of steps that we detected. Very few of these devices actually captured 500 steps spot on. Most of them had a substantial amount of underestimation. And then when you look at them and you look at the error bars around these mean steps, you see a lot of variability. That is, not only are they missing the mark, but they have a lot of imprecision for these individuals. The only device that actually worked quite well was the Fitbit 1. I have no stock in Fitbit. I have no benefit to showing that this device works. But this is the only device that had low error and high precision. Yes, ma'am?
You might be getting to this point, but what about the Hawthorne Effect movements? Because they have biofeedback?
Yeah, so in this context, we didn't have to worry about this because this is a controlled environment where we had individuals do a specific number of steps on our treadmill. So they're not self-reporting this or recording it. And we're not trying to change their behavior in this context. But absolutely, with these devices, when you put a device on someone, they know they're being monitored, right? And so one of the keys is figuring out how long you have to monitor someone to get a stable response that doesn't reflect reactivity. And
I have the answer.
How long?
Until they put it in the drawer.
Yeah.
Because that's where most of these end up.
And the question is why do they end up there?
Right. Right here.
Why do they end up in the drawer? Well, the battery dies, or they don't actually give you real, accurate, and precise data. And so this is a real issue. One, reactivity can matter, but the other issue is that if I use this device and it doesn't give me the data that I suspect I should be getting, what am I going to do with it? I'm going to stop wearing it. And we have firsthand experience with this. We use these devices in interventions all the time. And people will call us up on the phone and say, so I walked for an hour today and I got 10 steps. What do you think happens to that device? It goes in the drawer. We work with them to figure out how to actually make it work right, but for a lot of individuals, these devices don't work right, particularly when people walk with a slow manner, with very little displacement of the center of mass and things along those lines. Did you have another question?
Wait.
Yes.
Yeah, I had 2 questions.
3.
How do you know that it's not giving them the feedback that they want? And how do you know it's not motivational?
So in this context here, but in general, how do we know it's not motivational? And so we actually use these as a motivational tool, right? So we're going to come to that in a second. That is, can you use technology as a motivational resource, once you get beyond reactivity? And we can get beyond reactivity. The real answer is you just need to monitor people like with physical activity for about 7 days, and then throw those 7 days away, and then start monitoring them after that.
I think
Yes.
I just want to follow up really quick. I think I think Rob was right. One of the reasons they end up in the drawers is because people get frustrated with the data maybe. Another reason is they have in their mind a perceived target that they're not reaching, and they just get tired of not reaching it, right? Oh, HHS tells me 10,000 steps, which, by the way, there's no evidence base for, but that's what we all say, right? And I'm tired of getting 7,200 steps. I just don't want to see myself failing all the time. I just take it off.
Yeah.
Are we meeting their expectations by having it on?
Right.
Well, and so a lot of what we do is in these types of interventions that use these sorts of devices, we help people understand what sort of realistic expectations they should have up front. So they're not thinking, well, I need to get 10,000 steps per day. So we teach them how to use it, but also give them realistic expectations about where they are and where they should be, if that makes sense.
Just an aside, you just analyzed data, Fitbit data on total hip arthroplasty patients, and we had a 21-day follow-up.
Yeah.
And they linearly increased up to the time we got Fitbits back. There was no plateau shown, and it was like perfect.
Yeah.
So it seemed like they were responding to their needs.
But maybe they're just continuing to get better and able to engage in more ambulation, right? No, that could be an alternative explanation.
Yeah.
They keep getting better. But, and that's the value of having a control condition at that point in time, that you can compare what is their reactivity against. So the other thing is what do the actual data that you're collecting mean, and can you link them with a biological substrate? So this matters because we want data, and we want to be able to understand what those data mean. And this is an example of a group of researchers who were looking at linking step rate accelerometer data with energy expenditure. And it's really important not to just trust default values in this world. So the previous notion is that in this group of individuals with MS, you could just apply an actigraph accelerometer and utilize the preconditions within the software program to process the data into time spent in different intensities of physical activity. Well, it turns out that that's very poor logic, because people with neurological diseases, in this case MS, don't walk the same as the general population for which those cut points were developed. And secondly, when you look at them, they actually walk at a higher energy expenditure for any given walking speed. So in this context, what was found is that the relationship between activity counts and energy expenditure as a biological substrate existed in MS just like it did in any other population, but you had to create new methods of processing and interpreting the data for MS. This probably is the same case in Parkinson's disease, in spinal cord injury for sure, and things along those lines. So you have to start with a sensor. The sensor is going to give you some data about an individual. The other thing you have to have is some sort of remote interaction platform for the exchange of information. And there are lots of opportunities out there. So, for example, you can use the internet. The internet is a wonderful tool to deliver information to different populations of interest. As an example, and I'm going to come to multiple sclerosis, people with MS are huge users of the internet. In fact, about 95% of people with multiple sclerosis say they use the internet to get health-related information about managing their disease. And they're actually the highest users of the internet of any population in the United States. And so knowing that, researchers have gone in trying to develop interventions that you can deliver resources to individuals through the internet to get rid of that need for people to come into a facility. And that's worked quite well. There are some other avenues as well, such as Skype or Zoom. That is, you can engage in one-on-one interactions in an individual's home environment using these sorts of mechanisms to where you can deliver information from afar into an individual's house. So that becomes very important. You also need remote delivery equipment if you want to do exercise training studies. And I think that these sorts of pieces of equipment are becoming more readily available, maybe a little less expensive. So this is an Espresso Nuvo cyclergometer. And the beauty of this ergometer is that it actually an electrically braked cyclergometer that has the internal workings consistent to what Marcas and I might use in our research laboratories for doing high quality exercise testing. But the benefit of this is that you can put it in someone's home and it connects to the internet. And it stores the data in the internet. So now we can prescribe exercise for someone to do in their home environment, and we can go into their device and see if they're actually doing what we've asked them to do at the right frequency, the right duration, the right intensity, and all of those characteristics. Now there is a caveat. How do you know it's me doing the exercise instead of my son? That is the one caveat. But more and more of these devices are getting more sophisticated in that you can put a camera on them and actually take a picture of the individual to determine who is actually doing the exercise. So now we have remote equipment delivery that it allows us for a very high fidelity intervention. And so cyclergometry might not be for everybody. This is FES cycling, so functional electrical stimulation cycling, where you can put electrodes on an individual's leg musculature, as an example in this picture, and have the machine electrically activate their muscles in a manner that produces a cycling exercise against a resistance. The beauty of this is that you can, again, teach people how to use this piece of equipment. It is FDA approved for home use, and it's wireless. So you can go in there and you can look at what they've done and very specifically pull the data. Another thing you can do from this, from afar, is you can actually adjust the exercise parameters for them. So if an individual is having problems, I can go in there from outside and change around the settings for their exercise stimulation and their exercise bouts. So now we have remote delivery equipment. Does anybody have experience with FES cycling? What about with one of these Espresso Nuvo bikes? These are becoming more popular in gyms as well because they have this interactivity component to them. And then you can put these all together, and this is from Scott Bickel, who's a colleague of Marcas and I, into a complete system. So here we start with a monitoring system. This is a Zephyr bioharness that allows you to control or measure heart rate data, breathing, respiratory data on an individual. We can link that up with a system for monitoring. Through the Wi-Fi, we can get internet access so that we can actually interact with an individual about their biological signals when they're exercising. We can then put that into a system where we have a remote delivery exercise stimulus, as we have here, where an individual has the iPad next to them. We can monitor their exercise session in real time. We know exactly who is exercising, and we know the exact intensity of the stimulus and the parameters so that we can manipulate what they're doing and encourage them and get a high fidelity intervention delivered in the home environment. So there are some issues. Access and reach to underserved populations, such as the rural environment. So we're doing a very large trial in rural Alabama right now. Well, it turns out that getting internet access to everybody in rural Alabama is much harder than we ever thought it was going to be. So we're trying to figure out how to get over it. Because we think that internet access is so ubiquitous, right? If you have a cell phone, there are cell towers everywhere, the signal is reaching everywhere. Well, it turns out in rural Alabama, that's not the case. Technological and usability issues. So older adults are cognitively impaired individuals. So we have a large group of older adults who didn't really grow up with this technology. And they're a little squeamish with it if you try to get them to utilize it. With the proper training, you can help them be able to utilize it very effectively. But then we have other individuals, such as those who are cognitively impaired, that we have to teach how to do this and hope that they can do it. The comparability with standard delivery, that is, we need more of these trials that look at traditional supervised versus home-based programs to be sure that we're getting the same effect at home that you do in a supervised facility. And this really will involve feasibility, pilot testing, efficacy, effectiveness, and looking at responders. And then there's this issue of whether or not this sort of stuff will ever be reimbursable or the licensing of technology. And I'm done, so all the questions you want.
I have a couple of other limitations with it. We have a trial going right now, the tele-rehab or telehealth study. And one of the problems that we're having is with licensure. Where it's a multi-site trial, and different states have different licensures on even if you can do telehealth, and then you have to do it within your own state. But additionally, the problem that we're having with exercise and getting it approved with the IRB is that we don't have defibrillators at their house. We emergency contact phone numbers and information every time we exercise with them. And then we've had to lower the intensity of the exercise just to get it approved with the IRB. So we've had to wash down our effect because we're not immediately present when we're doing it.
Yeah, and we've encountered similar problems when you start trying to do multi-site studies across states, not just delivering your intervention remotely, but also collecting your data remotely. Some places and universities don't allow for kind of an electronic distance-based capture. They're fine with the intervention being delivered, but you want to capture data from them in their home environment. They're not okay with that, or they're not okay with the platform. So is it HIPAA compliant? Absolutely. And so as a good example, Skype is not HIPAA compliant, whereas Zoom or GoToMeeting is, right? But when you go talk to people and you say, hey, I want you to download the software for Zoom, they're like, what is Zoom? If you say Skype, everybody knows who Skype is right now. It's almost preloaded on every computer, right? So there are issues in moving past HIPAA-related regulations and things along those lines. I think we have to do more of this work to get more clarity. And we have the fortune of working with a lot of industry partners, and industry partners are really accelerating this space right now. So they are working very quickly to develop these very complete systems that can process data very, very well with a high degree of accuracy and precision and fidelity. And they're developing platforms that are HIPAA compliant for delivering interventions as well as monitoring them. So I think in the next, let's be optimistic, 5 years, we're going to have complete systems for doing this that allow us to do a much better job. Now, how do you get beyond the issue of safety in someone's home environment? That's, to me, that's a real education of IRBs, right? That's where IRBs need to be educated, i.e., you know, it used to be that age was an absolute contraindication to exercising, right? Nowadays, we've gotten rid of that because ACSM has gotten wiser. And IRBs are still stuck in the old age of if a male is 55, if a male is 45 and a female is 55, that's an absolute contraindication. It's just not true. So it's, as a former chair of the IRB, something that you have to educate IRBs on.
We're working on that. We have one institution that's approved and one that's not, one's in transition. It's a real issue. We've also had to make our subject population so homogenous. It's no longer pragmatic, and I worry about the external validity of it.
Yeah.
But it's a problem because I think this is a new area, and I do Parkinson's disease, and so they're all older, so the issues of age and heart conditions and things like that are really problematic.
And risks of falls.
That's
You know, you hear that quite a lot. How, you know, our first home-based internet-delivered intervention, our IRB just said, how are you going to make it so people don't fall? And it turned out no one fell. So it was a good thing later on, but it's yes, ma'am?
Yeah, well, no, we were like going for a grant for a similar idea, but one of our neurologists wanted to do a telehealth fall prevention program. And I was like, okay, so fall prevention exercise, the therapeutic dose is really close to the toxic dose if you want to get better. So yeah, I think that it was even hard just to like get through the door because it's just like, I don't feel comfortable actually doing fall prevention at home. Because again, what works, you know, is hazardous.
Yeah, putting people at the limits of stability to get them to adapt can be, yes.
Yeah, and also, is there an article with the figure of sources of variability? Was that a figure from an article, or?
Yeah, that's
Actually, it's adapted. It's because we've evolved it.
Oh, okay.
The original figure published in 2014
Okay.
is in Mayo Clinic Proceedings.
Okay, thank you.
And that's a paper that actually grew out of the national network I mentioned, NextNet. So that was several of us sort of thinking about this from multiple institutions. We've evolved it over time. The version that you'll see in Mayo Clinic Proceedings 2014, I'm the first author, is, you know, it's an exercise focus, but we've evolved this to capture more than exercise. We're sort of up against the hour. Are there any more? I mean, I'm not going to cut you off. We've got time, but are there any other questions?
My plane doesn't leave till 7, so.
You can gravitate toward the bar with Rob.
Well, no, my quick questions are this. What are the issues about safety and doing studies at home? And the reason why I'm asking this is because I do long-term care at home. I do home health care at home. And these are the kind of things that we actually want to do. And, you know, like study this with our patients in a real community setting and helping them improve their care. Because we have patients with, with chronic care that I'm trying to correct. So for me, because I know there's going to be a home health aide in the house, so why should I have any concern about what's going to go on? We teach them, you know, how to use the equipment properly.
Go ahead.
His question is, how do I maximize safety and minimize potential risks of delivering an intervention in someone's home? And so I always kind of approach that question from an IRB perspective. That is, identifying the situations that increase the risk of some event occurring, and then trying to do everything you can to identify a mitigation strategy. So for example, in the situation of someone who's doing a fall-based intervention at home, where we're putting them at the limits of their stability to try to get them to adapt, could we think about an approach to reduce the likelihood of them actually falling? And so I'm not a fall-based researcher, but I might think about, well, could I ask them to do this in a hallway within their house that allows them to, fall into the wall, not fall to the floor? Or could I have them do this next to a couch where they have their hands out next to them? Or do this with, if they're in a fortunate situation, have a partner at their house, do this within the confines of working with a partner. We know there are risks. Anytime you intervene, there are risks. The real question is, can you mitigate those risks? And so that's the way I think about it, is what are the risks, and then are there ways of reducing those risks? Does that make sense?
That makes perfect sense. But what I'm saying is, I guess, from my perspective, if I've got a home health care aide in the house, and this person actually needs balance training of some sort, and you have a program that actually works, well, why can't I use that home health aide to help you give her the balance training while she's there, so that the patient does it in a guarded environment.
And I think in an ideal sense, having someone there with the patient would be great, right? But there are lots of situations where we don't have a home health care aide there. Does that make sense? So in the context of having a home health care aide, I think doing it with them, teaching them, making sure they're doing it correctly. A lot of times we use a hybrid model where we're doing home-based exercise programs. We actually bring people into the lab on a somewhat regular basis and teach them and make sure they know how to do things and do things correctly, so they're reducing the likelihood of some event occurring, and then sending them home. Because a lot of situations we don't have that coverage of a home health care aide. Does that make more sense? So if you have it, great, use it. But if you don't have it, then we have to figure out a way around it.
There's also the advantage of, if you do have, Rob gave the example of the direct video monitoring, where it's as if you're there. So if there was a safety issue and you were actually interacting with them directly, it would almost be as effective as them being with you, if you can have a response to that. But that model of testing people in the home using audiovisual technology and biosensing was first done in spinal cord injury, and now it's been done in Parkinson's. And I think we're trying to improve on it, but I think it's a model that can be used for a lot of people who just don't want to navigate New York City, or it's not they don't want a 90-minute transit each way to go do this intervention study, right? As an example. So I think this is a wave of the future for not only some of what you heard about today, but I think you can apply this really broadly. We all run into these catchment area issues where we write the grant application and what's reasonable? 3 days a week, okay, 30-mile catchment radius. What's the diversity within my 30-mile catchment radius? Because if I go beyond that, I'm not going to get anybody. But now, with what you're talking about, you can go as far as you can go. And if researchers have to drive the availability of wireless, that's just part of the deal, which I'm sure that's what you're facing. But
so what other questions are there? Yeah. Any other questions? Yeah. Where's lunch? Or you already had lunch, sorry.
Yeah, we haven't.
We haven't. Any other questions?
So use REACT to your advantage, and also look at the other 5 centers and find opportunities to leverage what we have. That's what they're for. They won't continue to be available to any of us if we don't actually leverage them and add value back for, get the NIH a large return on investment.
And if you are writing an LOI for React,
Yeah.
there's a little component in there, how are you going to utilize React? Take that seriously and start interacting with React even before you put in your LOI. There's no reason not to set yourself up to be as successful as you can up front. Did you have a question?
I have a question about the adaptive study design. That usually in this kind of design, would we set a primary outcome, stick to that outcome, and provide several options or sequences or maybe dosage of different interventions? Or if we change the primary outcome or even the purpose of studies and tailor them together, would they still be considered a adaptive study design?
No. Well, it's highly unlikely that you would use adaptive design on the front end, predicting that you're going to change the primary outcome. It's the ways of testing the outcome that you're going to predict on the front end, or design into it on the front end. The interim analysis, the potential alternatives that you might switch to, the sample size analysis, all those things in the adaptive model are ways of getting at the primary outcome or the primary question most effectively. When you start fishing for a new primary outcome, it's not really a pre-planned adaptive. Any other questions to wrap this up?
Yeah,
Yes, sir.
I'm just wondering how you go about the process of interacting with React. Does it start off with a submission via the web portal?
It's web-based. So right now, if you want a consultation, you would go to the website and there's a drop-down. You click there, a little window you fill out. We're not asking for too much because we don't want it to be burdensome. Just a little description, which then when you submit that, it will enable Carla and others to know which investigator can best answer that or serve in that way. So if it's, molecular biology, it's coming to me. If it's, do I go adaptive, it's going to Gary Cutter. That kind of a thing. If it's, how do I incorporate this biosensor, it's going to Rob. So we have a really good diverse team, but that enables us to direct the traffic. And the policy is that you should hear back from us within 48 hours. And then if it requires a phone consult, then we have people, Carla has people working for her who will set that up.
Other funding opportunities, just so you know, drop down Scholar Awards, that's where you fill it out.
Well, that's yes. So Scholar Award is a term we use for a lot of things. We may, like I said, I gave you an example of we analyze samples for you. But if you consult with Rob for 6 hours, that's all good. Doesn't cost you a penny. We just have to account for it on our side so that we're being cognizant of the money that we've been awarded. So in that sense, you don't get a check. Hey, I got a $600 Scholar Award or whatever, $3,000 Scholar Award. No, but we accounted for that service, kind of a thing. But yeah, that's where you'd find the form. It's really easy. But hopefully we'll see some of you apply for pilots as well. And the one thing I didn't say, if you are a pre or postdoc, this wasn't intended to advertise our training grant, but we do have at UAB a interdisciplinary T32 program in pathobiology and rehab medicine. So both pre-doc and postdoc slots on the grant. Obviously, if you're a pre-doc now, you're in a program that's very good, but when you do look for your postdoc, just look us up. Okay? Thanks, everybody.